Yeah. Thank you. Ladies and gentlemen, we'll be starting the session in 1 minute. No, it doesn't give it away, does it? Good afternoon, ladies and gentlemen. My name's Bob Stevens. I am Group CEO for the MPS Society UK and MPS Commercial, and along with Terry Klein, Joint Chair of the International MPS Network. It gives me great pleasure today to introduce the session on next generation treatments, combination therapies, and advancing clinical trials. As an MPS 2 dad, I'm every bit as interested as everybody else in the room as to what does the future look like. So before our next speaker comes up, I will just say a few words on him. So Michael Beck was the head of Centre for Lysosomal Storage Disorders at the University of Mainz and is a genetic counsellor at the Genetic Institute of Mainz. And he has organised 2 international MPS conferences in Germany. So, Mr. Beck, if you'd like to pop up.
Dear Bob, dear family, at first I would like to thank you for the kind introduction, and also to thank the organizers, Stephanie and Patty Dixon, for having invited me to give a talk here at the family conference. For me, it is always a great pleasure to be an active part of the family conference at the MPS Symposia in the last years. I have been asked to give an overview about new treatments, and of course in 30 minutes it's not possible to cover all aspects and to go into detail, but I will give a short review, and in the next presentations you will hear much more about, we get much more detailed information. For example, gene therapy, that will be the next topics after my talk. And I will talk not only about your treatment options, what we can expect in the future, but also would like to show how much parents or the patients group can contribute to develop new treatments. I will give some examples. Everybody knows enzyme replacement therapy, and Dr. Bigger has already given or talked about the limitations of enzyme replacement therapy, and therefore I will go very fast through this slide. What can we expect from enzyme replacement therapy? Reduction of storage material, everybody knows that the glycosaminoglycan excretion goes down when you start enzyme replacement therapy. We have seen a reduction of organ size and also some gain of function of the general endurance that is measured by the 6-minute walk test, some improvement in the lung function, improvement in mobility, and also some improvement of the heart function. But one of the biggest limitations of enzyme replacement therapy is that the enzymes that are given intravenously are not able to enter the blood-brain barrier. How can we overcome this problem? One is to give the enzyme intrathecally, what has been done in single cases, but has also been done in a clinical trial. And this results have been published 2 years ago by Joe Münzer. And what has been observed, it was done in some patients with mucopolysaccharidosis type 2 Hunter disease. And what one has been seen, that indeed by giving the drug intrathecally by using a special device, that you can really see a reduction of glycosaminoglycan concentration in the intraspinal fluid. Here, very short reduction within some weeks after the application. But they are going on other clinical trials, but in this study, the neurocognitive evaluation was not performed. Therefore, one cannot say anything of whether this procedure has improved the cognitive abilities of these patients. Other clinical trials have been performed also in Sanfilippo disease, for example, and are still going on. Another way is to modify the enzyme that makes it able to cross the blood-brain barrier. And for me, one of the very new way and also possible way to do this is by using antibodies that are bound to the enzyme. I will go into the detail a little bit more. And the scientist who has developed this method, Boado, called this Trojan horse brain enzyme delivery. What does it mean? That means he has bound to the enzyme. He calls it Trojan horse, but indeed it's an antibody against insulin receptor. And by binding this component to the enzyme, this complex can be taken up by the insulin receptor that is in every tissues and also at the blood-brain barrier. And then it can pass the blood-brain barrier, and after passing also this complex can also go into the cells, and the enzyme can be active, degrade the substrate. And this below shows without modification, the enzyme cannot go into the brain. And how does it look? And has been clinical trials performed. This is how this Trojan horse, or this, it is called fusion protein, is composed. There's the enzyme, in this case it's iduronidase, also the enzyme that is deficient in MPS 1, and this is bound to an monoclonal antibody against the human insulin receptor. And this complex, it has been shown that this complex is catalytically active. That means is able to degrade glycosaminoglycans. And of course, before you do any clinical trial in humans, it has been analyzed carefully in animals, and this shows that this complex of enzyme and antibody indeed is able to go into the brain, and this is shown by special spectrometry in monkeys. And these monkeys have got this Trojan horse, this complex fusion protein, intravenously, not into the brain. And after some time, it could be shown that indeed it is taken up by the brain and is everywhere into the brain structure. All the red one points shows the active enzyme. And after this very promising animal study, one has done also a clinical trial, and these results have been published just recently in this year. And it was a 26-week study in patients with MPS I. These patients had the age of 2 to 15 years, and one has seen really, this was given intravenously, and one has seen that this drug that is called RGT-181, that is the fusion protein of hyaluronidase and insulin monoclonal antibody against the insulin receptor. And this drug was very well tolerated. This patient had already treated with enzyme replacement therapy many years ago before. That means they have got this after enzyme replacement therapy, and therefore stabilization of urinary gags. They didn't go up again. Improvement even, although they had already a long time enzyme replacement therapy, there was an improvement in shoulder motion, stabilization of liver spleen volume, stabilization because it was already a reduced size because of the enzyme replacement therapy with the enzyme many years ago. And on a cognitive level, interim data, these are not conclusion, in general conclusion, what one has observed, improvement in neurological or cognitive function in 4 of these 5 patients, stabilization of neurological function seen in 1 patient, and of course, more data have to be collected to really show the efficacy of this new treatment. Another way to treat for treatment, of course, as you know, is substrate inhibition. And since a long time, genistein has been shown that it's probably able to reduce the glycosaminoglycan concentration. Genistein, as you know, is a component isoflavone of the soya bean, and in cultured skin fibroblasts, one has shown that this component is able to reduce glycosaminoglycan synthesis. And in patient, the Polish group, the Dutch group has shown that indeed, by giving the genistein, the glycosaminoglycan excretion goes down. And there has been performed a clinical trial with high dosage of genistein in Manchester, but I don't know, have not heard anything about the results of this clinical trial. There was a clinical trial with 19 sanfilippo patients. It has been published many years ago. And the dosage was rather high. There was no reduction of glycosaminoglycan excretion, no improvement of cognitive function. Let's see what the Manchester clinical trial will show. That's a way of substrate inhibition as a treatment. Then I come very shortly to gene therapy, because you will hear much more about in the next presentations. Gene therapy is, one has spoken about gene therapy in mucopolysaccharidosis and lysosomal storage disorders in general since more than 20 years. And when I started in this field, everybody talked about very soon we will have gene therapy, but for me it seems that it's indeed the case that we can expect, and already the first clinical trials have been performed and will be performed. Why are lysosomal storage disorders in general, not only mucopolysaccharidosis, are excellent candidates for gene therapy for several reasons. They are very well characterized single gene disorders, and you don't need 100% for normal function. You can see it in heterozygotes, the carriers of the disease. They have about 50%, but it has been shown in many lysosomal storage disorders that 15 to 20% is sufficient for clinical efficacy. And it is enough to bring the gene in one of the organs, for example, an organ, and the organ will secrete the enzyme, and it can be taken up by other cells. But of course, if you bring the gene into the liver, it will secrete the enzyme, but of course also this enzyme will not be able to cross the blood-brain barrier. It's the same, more or less, principle like the intravenously application, but you have to do this gene therapy, of course, only once. And one of the very essential aspects is also there is no complex regulation necessary. It's enough if you have 20 or 25% of the enzyme. There are two methods to bring the gene into the body. One is the ex vivo therapy. That means that you collect stem cells from the patient, bring them in culture, and in the culture you bring the gene using a vector, a viral vector, into the cells, and these modified cells were brought back to the body. And this method has been done several years ago in Italy in the lysosomal storage disorders metachromatic leukodystrophy, and the results were really overwhelming. And this has been done, it was very essential to understand. It has been done in 3 children with metachromatic leukodystrophy who have not been any symptoms. That means they were asymptomatic when they started this gene therapy. And only 3 patients have been selected for this clinical, for this trial. And this shows the results. This is a measurement of the gross motor function that is reduced in these patients. And all these points are patients with a natural history. That means patients with metachromatic leukodystrophy without any treatment don't have any gross motor function. But these patients are able to move absolutely normally and are comparable to normal individuals. And these are the results of 2013. And some years later, another paper came out from the group from Dr. Biffi in Italy, and they showed that even after many years, the 3 patients are normally from the movement and also in cognitive aspects. That means this example shows that indeed ex vivo gene therapy seems to work. The other way to bring the gene to the body is in vivo gene therapy. That means that the gene is directly brought into the body. And you know, the Tardieu in France has been done this in San Filippo A patients. And there's another, one has seen that the results have not so successful as one has expected. Therefore, a group from Dr. Chen has modified the sulfamidase that is deficient in San Filippo A, and he has shown in an animal model, in a mouse model of Sanfilippo A, that indeed by modifying this the sulfamidase, gene therapy is more effective. That has been shown by measuring the enzyme activity. These black are normal controls, and these gray one, these dark gray are the animals who have not been treated, of course, with a very low enzyme activity. And the white one was by using the normal sulfamidase and the AAV vector. Of course, you need also for gene therapy this vector. And of course, this increases the enzyme activity. But if you modify this sulfamidase HV4, then you can even more increase the enzyme activity. And of course, one has not only looked for the enzyme activity in several parts of the body, of the brain. It's the hippocampus, striatum, occipital cortex, and cerebellum. But one has also seen improvement in the function. And one of the methods to measure the movement and cognitive function in mouse is measuring the distance the mice can walk. And this is the unaffected mouse. These are mice that are treated or have received gene therapy. And you can see that mice who are treated with a modified enzyme can even much more walk than the mice that have been treated with unmodified enzyme. One has seen in more animal study whether it's really helpful to also modify the sulfamidase. And what is very surprising, I heard last year the first time, that one group has looked whether you can apply the enzyme intranasally, and was done, of course, only in an animal study in the mouse model of MPS 1. And one has seen this hyaluronidase was given intranasally and was taken directly taken up. But not only by the brain, but also by other organs, as you can see from the results here. That's the enzyme activity, and this is the enzyme activity of the affected mice. That is the enzyme activity in heterozygotes, and that were 6 mice. And you can see that by giving this intravenously, intranasally, also as the gene therapy intranasally, that you can indeed see an increased enzyme activity. Whether this method ever will come to the clinical trial, I don't know. And everybody has about the CRISPR method. What does it mean? This shows a scheme of this method that it makes it able to modify the gene in rather easy way, and has been found this method some years ago. And what does it mean? Here you see the DNA double strand, and this Cas9 is able to cut the DNA at different positions, and to find the right position you want to have, you need a guided RNA, and this complex is called CRISPR-Cas9 method. You can see this Cas9 cut the DNA, and after cutting you can bring another DNA or delete it, whatever you want, and then you have a controlled gene modification on a position you really want to have. And this shows is in the same method how could it be done. You have a patient. These are the human stem cells, that are modified by using the CRISPR method, and then the corrected stem cells are brought back to the patient. That means from the principle is the same way as has been done in the patients with metachromatic leukodystrophy, but the method how the gene is modified is different. Doesn't work. And the next one, please. Can you do it? Yeah. There's a new therapeutic principle. It means that are using as a combination therapy anti-inflammatory drugs. In the last 10 or 15 years, one has more and more detected that not only in mucopolysaccharidosis but also in other lysosomal disorders, inflammation plays a very essential role in the pathogenesis, and also in the brain of Sanfilippo patients, one can find increased inflammatory substances, cytokines, and so on. And therefore, the idea came, why not to give an anti-inflammatory drug to improve the situation for the patient. And that it really is essential has been studied also in animal models. I will not go into the detail. This is a group. There was also a presentation from Dr. Simonaro and Schuchman and so on. And they discussed that an anti-inflammatory drug that is in Europe, in Germany, is on the market, but not for mucopolysaccharidosis, may help the patients. And therefore, one has done a very small study at the Villa Metabolica, where I was the head before, and it has been done by my successor, Professor Hennemann. She took 4 patients with MPS I, older patients who have been treated with enzyme replacement therapy since a long time with different dosage. You can see that indeed by using polypentosan, the glycosaminoglycan excretion went down, although these patients have been under enzyme replacement therapy for a long time before. The next one. But there was not only reduction of glycosaminoglycan excretion, but some of the 4 patients really had less pain. That's not surprising because this drug is an anti-inflammatory drug, and many other clinical trials are planned to really confirm this effect on the clinical manifestation of pain and maybe other symptoms of the patients. I have not much more time, I see. Just very short, which importance have the parents group, advocacy groups for development of new drugs? And I will show very shortly 3 examples. One of the first examples, I will go not through. The first examples was the development of enzyme replacement therapy for MPS I. What was the first one for where this treatment is available? And this development of this drug was made possible by the Rhein Foundation. Rhein is the father of an MPS I patient. He collected many money, and Emil Kakis and Elisabeth Neufeld have developed the drug, and it came on the market for the treatment of patients with MPS I. And there's another Jonas Bass association. Jonas just begun. Founder is Jill Wood, a mother of an affected patient with MPS IIIC. And this family has collected money, and they founded a company, Phoenix Nest Incorporation, and this will develop in chaperone therapy for mucopolysaccharidosis type 3C, because, as you know, the enzyme replacement therapy will never be possible because this enzyme is membrane bound. And another goes through. And another very famous example is the company Lysogene. This has been founded by a mother of a San Filippo child, Ornella, and she also collected money, and they founded the company, and this company has performed the first clinical gene therapy trial. And this shows that you have a really very essential role in the development of new therapeutic options, and it's in your hands to go to the government, to scientists, to companies, and to collect money to make the treatment possible, not only for that is presently, but also to make treatment possible for the future. And with this, I would like to end my presentation. I'm open for questions if it's time for this. Thank you very much.
We've got time for a couple of questions from the floor. Are there any questions for Dr. Beck?
Okay.
Thank you very much.
If you have questions, you can ask, talk to me. Yeah. Thank you very much.
Okay, our next speaker is Mark Sands, PhD, and Mark is a professor in the Department of Internal Medicine and Genetics at Washington University School of Medicine. Over the past 30 years, Dr. Sands' research has focused on stem cell modified or medicated therapy, and the primary disease that Dr. Sands studies are mucopolysaccharidosis type 7, Krabbe disease, and infantile neuroceroid lipofuscinosis. Why do they make the disease name so long?
Batten disease. Well, thank you for the kind introduction. Thank you. So what I'd like to do today is basically provide a very fundamental and basic explanation of what gene therapy is, what the goals are we're trying to accomplish, and from a sort of superficial technical perspective, how we go about doing that. I'm going to break the talk up into several parts. First of all, I'm going to review autosomal recessive diseases and cross-correction, which, again, will be a review for everyone here. But there's a couple important points I'd like to emphasize, which will make sure that we're all on the same page when we talk about the gene therapy technology. And then we'll dig down into the weeds a little further on exactly what gene therapy is and how we accomplish it. And then at the end, I'll give a very brief summary of some of the strengths or some of the potential of gene therapy, and still some of the challenges that face us with respect to this very promising approach. So, autosomal recessive diseases. Again, this is a review for everybody in the crowd, but again, I want to make sure we're all on the same page when we dig down in the weeds a little further. There it is. So, these circles up here represent cells from either a man or a woman, and the dark lines here represent chromosomes or a gene, and the X here indicates a gene that's not functional, okay? And in this case, the male and the female are carriers. So what I mean by that is one gene is functional, and again, as review, we all know that we have 2 genes for every single protein or every single trait. In the case of a carrier, one gene is functional, the other gene is not functional. It's the same with dad and mom. Now, if mom and dad decide to have kids, and again, for review, you would have a 25% chance of your child inheriting a functional copy of the gene from mom and a functional copy from dad. Okay, clearly, this child would not have the disease. It would also not be a carrier. It could not pass on the non-functional gene to future generations. You'd have a 50% chance of your children inheriting a functional gene from mom or dad, and a non-functional gene, again, from mom or dad, okay? This child would also not have the disease, but it would be essentially carrier status. So it could pass this non-functional gene on to future generations. Unfortunately, you'd have a 25% chance of a child inheriting a non-functional copy of the gene from dad and a non-functional copy of the gene from mom. Of course, this child has a disease, in this case, one of the mucopolysaccharidoses. One of the points I want to make clear, though, at this juncture is, again, this is all review for you folks. You live this every day. But the one point I want to make with respect to gene therapy is these two non-functional genes, with respect to a gene therapy approach, they're not doing anything bad in there. They're pretty much benign. They're just sitting there, okay? So from a gene therapy's perspective, we don't have to get rid of or fix those genes. They're not doing anything bad. They're just not doing their job, okay? So the whole idea of gene therapy is to, or at least in one scenario of gene therapy, is to sort of ignore these, but try to add in a functional copy of the gene, okay? So conceptually, gene therapy is really very simple. You just want to add a functional copy of the gene in there so the patient's own cells can make the enzyme. It's really, again, conceptually, it's a very easy concept. So, cross-correction. Again, this is a review for everybody in the crowd. And just as a very quick review, in this cartoon, if this is a genetically modified cell here, okay, this cell is now making its own enzyme, it's correctly processed, it goes to the lysosome, but lysosomal enzymes have this unique characteristic that although they function within a cell, and in fact within a membrane-bound vesicle inside the cell, some proportion of lysosomal enzymes are secreted. And when they're secreted, they can be taken up by an adjacent cell, what I refer to here as an enzyme-deficient cell, so a non-genetically modified cell. Again, as you all know, this is the principle behind enzyme replacement therapy. So why am I bringing it up with respect to gene therapy? The reason is, with the current state of technology, we do not have the ability to genetically modify every cell in the body. And as you all know, with these diseases, every cell is affected. But again, we do not have the technology to modify every cell. So what we have to count on is cross-correction, where we can genetically modify a small number of cells in the body or in a particular tissue. Those cells can then overexpress the enzyme, and through cross-correction, biochemically correct adjacent cells, okay? So again, it's not just for enzyme replacement, it's also for gene therapy. So I want to make sure that those points are very clear. So what is the overall goal of gene therapy? Again, conceptually, it's extremely simple, okay? You've got two non-functional genes. They're not doing anything bad. They're just sitting there. So what we want to do is add a functional copy of the defective gene. And we refer to that as gene replacement therapy, okay? We're not correcting the gene. We're just putting a functional copy in. Alternatively, with new technology that Dr. Beck touched on briefly called gene editing, we now have the potential. It's not in the clinic yet, and the technology is really in its infancy. But there is the potential now that in the future, we may actually be able to go in and correct the exact defect in the gene, okay? Again, it's in its infancy. There's no clinical trials yet. But on the horizon, this may be possible. And again, there's gene replacement, which is currently moving into the clinic now, and gene editing, which may come to fruition in the future. And again, overall, what we're trying to do is we're trying to make the patient's own cells create the enzyme. So in other words, instead of bringing your child to the clinic every week or every other week and have enzyme infusions, if this gene therapy approach works, the patient will make their own enzyme. And if it works really well, they'll make their own enzyme for the rest of their life after a single treatment. So that's really the power of gene therapy. That's the potential. Okay, so if we compare and contrast gene therapy and enzyme replacement, I'm showing this is a completely hypothetical graph, okay? This shows enzyme levels, this shows time, all right? If you look at these two blue lines, again, hypothetically, this represents sort of the lower level of normal enzyme activity, and this shows the upper level. And of course, everybody in this room, we're all different. We have slightly different levels of enzyme activity, but this represents, again, hypothetically, the normal range of enzyme activity. If you do enzyme replacement therapy, this is what your enzyme levels look like over time. If you get an injection of enzyme here, you have a very rapid increase in enzyme activity that probably in certain tissues can exceed normal levels. And then as time goes on, the enzyme goes away. You now go below normal levels. You probably even go below therapeutic levels. Okay? Then you get another injection. You get this peak of activity, it declines, peak, decline. And this cyclic pattern is what you're dealing with for the rest of the patient's life. Okay? Now, in contrast, what we're trying to accomplish in gene therapy is basically to administer the gene therapy at some point in time, and then you get some level of enzyme activity, and then that enzyme activity persists. Again, if in principle, if this works really well, the enzyme activity persists for the life of the patient, and it's a relatively stable amount of enzyme activity. And if we're really lucky, it falls somewhere within this normal range, or at least within a therapeutic range, which, as Dr. Beck alluded to, could actually be below what we consider normal. This is what we're trying to accomplish compared to what's available right now. Okay? So in principle, again, it's where we want to be. So, what about gene therapy? So the first topic I'm going to cover is gene replacement therapy. Okay? And some of us have been working on this for a very long time. I know I've been working on it for about 30 years now. And it is finally starting to come to fruition. Okay, so what is gene replacement therapy? So I'm going to use this similar cartoon to what I used before. And again, in gene replacement therapy, we're not trying to correct the defective genes. Again, they're not doing anything bad. They're just not working. So what we'd like to do is add a functional copy of the defective gene. And if everything goes well, this potentially would provide a permanent source and hopefully a therapeutic level of the deficient enzyme. All right? So schematically, what this looks like in this cartoon, if this is the diseased cell, so you have two non-functional copies of the gene, what we need to do is add a functional copy of the gene, and we use viruses to do that. And then what the cell looks like is you still have these two non-functional copies of the gene. Now you have a functional copy in there as well. So at a minimum, you now have 3 copies of the gene in a cell. 2 don't work, 1 does work. Okay? So probably asking the question, why do we use viruses to do that? On the surface, this sounds like a really bad idea. Because if you think about it, in the most benign cases, the viruses, they give you a snotty nose, you get a cold. But many viruses will kill you. Okay? Why are we using viruses to do that? So the way my tiny little brain thinks about this problem is, if you think about a small molecule drug, and let's take aspirin, for example. Everybody uses aspirin. If you got a headache, you take aspirin, aches and pains, arthritis. It's almost a perfect drug. It does everything. It's a great drug. But if you look at the size, if you look at the molecular weight of aspirin, it's about 180 Daltons, okay? Don't worry about the Dalton part. That's just what us nerd geeks use to measure these things. So it's about 180 Daltons. Now, if you think about an expression cassette, so if you think about this functional gene that we want to put into a cell, even if I were able to cut it down to a very small functional gene and have DNA sequences on either end that help regulate it, which are required, the smallest expression cassette I can come up with has a molecular weight of about 3 million Daltons. Okay, so think about that for a minute. This expression cassette, so this piece of DNA, this functional gene I want to get into the cell, is about 17,000 times larger than something like aspirin. Okay? It's very difficult to get it in the cell at a molecular level. And even if you're able to get it in the cell, it's a very treacherous journey from the cell membrane into the nucleus. There's lots of places along the way where the DNA could be degraded. It might just sit there, or it could be directed to a completely different compartment besides the nucleus. So this is really hard to do. So what we do is we've taken advantage of the mechanisms that viruses have evolved over millions of years to get their genetic material into a cell. So the whole goal of a virus is to get its genetic material into the nucleus of a cell. so that it can produce its proteins, so it can produce baby viruses, which then go on and infect cells, and you get this cycle that goes on and on and on. And viruses are very efficient at it. So the way we do this is we take these viruses and we break them apart. We then take out the viral genes that cause disease and also the viral genes that cause them to make baby viruses. We take those genes out, we throw them away. Then what we do is we take a functional copy of the gene that's defective in whatever disease we're looking at, and we put it in place of those viral genes that we've thrown away. Then we take those viruses and we put them all back together. And now you've got a virus. We actually, those of us in the field, we oftentimes call them suicide viruses. You've now got a virus that will infect a cell. It will transfer the genetic material to the nucleus where it's fully functional. But because we've thrown away all the disease-causing genes and the genes responsible for replication, you don't get a disease, you don't get baby virus particles out at the end. All it does is transfer the genetic material, in this case the functional gene, to the nucleus. Okay? This is just a very short list of some of the viruses that people have worked on and developed them as gene therapy tools. We've got adenoviruses, adeno-associated virus, retroviruses, lentiviruses, herpes viruses. And this is a very short list. There's lots of viruses that people are manipulating to use as gene therapy tools. Okay, so I'm going to refresh your memory on some of the stuff Dr. Beck just talked about. In this case, there's multiple different methods of gene therapy, one of which is ex vivo gene therapy. And to put it very simply, what we do is we remove the patient's own stem cells. Okay, so a stem cell is something that if you take it out and put it back in, it can find its home, it can make more cells, and it, in principle, can persist for the entire life of the patient. You take these cells out of the patient, you put them in a dish, and then within that dish, you put these viruses on. The viruses infect the cells, they transfer the functional gene to the nucleus. You then take those cells, you wash off all the excess virus, you transplant them back into the patient. And if everything goes well, these genetically modified cells can produce and share the enzyme through cross-correction, hopefully for the life of the patient. Now, the most common stem cells that we like to use are hematopoietic stem cells. And these hematopoietic stem cells, these are stem cells that reside in the bone, so your bone marrow. Okay? The bone marrow gives rise to your blood cells. And if you think about this for a minute, if you take out the bone marrow stem cells, the blood stem cells, and you genetically modify them, they're now producing the enzyme. If you then transplant those cells back into the patient, now the patient's circulating blood cells are corrected. They're expressing the enzyme. And they, as they circulate around the body, they can share that enzyme. So if you think about that, it's a wonderful delivery mechanism. Blood has access to every tissue, and if these cells are through cross-correction, if they're sharing their enzyme, at least in principle, you might be able to get a very effective and more complete therapeutic outcome. But also, if you think about it, as Dr. Beck mentioned, we have this pesky thing called the blood-brain barrier, which is very effective at keeping enzyme out, it keeps other cells out, it keeps viruses out. So you could potentially think about taking neuronal stem cells, taking them out, modifying them, putting them back in, or mesenchymal stem cells, or pick your favorite flavor of stem cell, liver, pancreas, whatever. Potentially you could take some out, modify them, put them back in, okay? The viruses that are most commonly used for that or most effective for this ex vivo approach would be the lentivectors or the retroviral vectors. Okay. The other approach is direct in vivo gene therapy. And what do I mean about that? This procedure basically cuts out that middle step. So instead of taking cells out of a patient, modifying them, and putting them back in, we're going to deliver virus directly to the patient. So again, you're cutting out that middle step. Okay? So you can inject the vector directly in the patient. Then you're genetically modifying the cells inside the body, so in vivo. And then, of course, through cross-correction, you like those cells to secrete the enzyme and hopefully get a very good therapeutic response. So what about the route of administration? The easiest route that you can think of is intravenous. You find a vein, you inject the virus, it infects cells, you deliver enzyme, okay? But again, as we talked about before, this pesky blood-brain barrier problem. If you inject the virus intravenously, it's not going to get in the brain. And as Dr. Beck mentioned as well, the enzyme that those cells are producing will not get in the brain either. So you could potentially inject the virus directly in the brain. Alternatively, you could do an intrathecal injection, so basically through a lumbar puncture, administer virus that way. If you wanted to treat the eye disease, you could think about injecting virus directly in the eye. And I'll tell you, all these experiments have shown efficacy in animal models, all these routes. And again, think about any tissue, any organ system. If you can gain access to it, which you can in most tissues, you can deliver a virus there. Right now, the darling of the gene therapy field are adeno-associated viruses, okay? They're small, relatively benign viruses, and they're very, very efficient at this in vivo gene therapy approach. Okay, what about clinical trials? So I went to the trouble to look on clinicaltrials.gov, and right now there are over 50 clinical trials listed for lysosomal storage diseases, okay? And of course, the mucopolysaccharidoses are a subset of lysosomal storage diseases. So of these 50 clinical trials, 15 of them are listed for MPS disorders, okay? One of them is an ex vivo gene therapy clinical trial, again, as I described before. And this targets the bone marrow, so hematopoietic stem cell directed. It's for MPS I, using a lentivector, and this is being currently performed in France. With respect to in vivo gene therapy, you can see there's a number of MPS disorders that are being, where there are clinical trials. MPS I, II, 3A, B, and MPS 6. The common denominator here, they're all using adeno-associated viral vectors, okay? And some of these are being performed in the US, some of them in other countries as well. So, that's the basics of gene replacement therapy, okay? Using standard vectors that some of us have been trying to develop for over 30 years. Now, what about the new technology, gene editing, okay? And there's two forms of gene editing I want to talk about. One is gene replacement and the other is gene correction. So with respect to using gene editing and replacement, again, very much like gene replacement therapy that I just talked about, we're not correcting the defective gene. We're simply adding a functional copy of the defective gene so that the cells can produce their own enzyme activity, hopefully permanently. What's different is what we're trying to do with gene editing replacement is put a functional copy of the gene in what I'll call a safe place. So diagrammatically, it looks like this. This is the diseased cell. Now you need to have several viruses to accomplish this goal. And this represents another chromosome here, and you're trying to put this functional copy of the gene, again, in a safe place. So just focus on the top and this side of this flow diagram. The one way to do this is using a technique called zinc finger nucleases. And remember I said you need two viruses to do this. One virus will encode for these zinc fingers, okay? And what is a zinc finger nuclease? Zinc fingers are these protein domains that you can very specifically engineer. When you engineer these, what are called zinc fingers, and you express them, these zinc fingers can actually scan the entire genome. They scurry around and they look for sequences that they like. And they will bind very, very specific sequences in the DNA. The other part of this zinc finger nuclease is, in fact, this nuclease, which I'm depicting here by scissors. So the zinc fingers find a specific spot. The nuclease cuts the DNA, okay? Now, cutting DNA is not a good thing, I'll tell you right now. But we have very efficient repair mechanisms that can correct that. Well, remember I told you also there's two viruses. The other virus contains a copy of the functional gene. It's referred to here as a repair template. So when you cut this DNA and then your body wants to repair it, oftentimes it will take this piece of repair DNA, which now has the functional gene, and it will put it in that spot, okay? And then you get something that looks like this. You insert this, or you replace this functional gene. And we know enough about the genome. We're still very ignorant about the entire genome, but we know enough about it that where there are safe places or safe havens where we know that we can make a cut and put in a foreign piece of DNA and not cause some other problem, okay? So, why use targeted gene replacement therapy? Well, if we take a couple steps back and talk about the gene replacement vectors that I talked about earlier, we're using viruses, and with some frequency, those viruses will integrate or insert into the patient's genome. When that happens, that's a new mutation. And we all know that mutations might be bad. Some of them are benign, but some of them can be bad as well. And these vectors might insert in many different places in the genome, and these mutations, so this insertion of this functional gene now, could either activate or inactivate a different important gene, causing yet another disease on top of the disease you're trying to treat, okay? So that's a real potential for the gene transfer vectors that I talked about earlier. So the targeting vectors, again, we're trying to, instead of having these things jump in the genome randomly, we're trying to make them go into a specific spot. And we can choose that spot, okay? And we know it's a benign spot because we've scanned the DNA around it. So we call those safe harbors, okay, or safe havens. If we can insert into that, we know this won't cause some secondary disease from the mutation, okay? And it reduces the chance of harmful insertions other places around the genome, okay? So that's using gene editing to replace a functional copy of the gene, okay? And there's actually 2 clinical trials ongoing now using this zinc finger nuclease-mediated technology. One is for MPS I, one is for MPS III. These are also using AAV, adeno-associated viral vectors with zinc fingers. It's delivered intravenously, and these clinical trials are going on in the US right now. So the final thing I'd like to talk about is gene editing, but in this case, actually correcting the non-functional genes. And of course, from a gene therapy's perspective, this is the holy grail. If we can simply correct the mutation that's there, that would be the best alternative, okay? So, in this particular case, unlike the other 2 cases I pointed out, we're not adding a functional gene. We're actually correcting the defective genes, okay? Again, the same thing with all the gene therapy approaches, potentially providing a permanent source of the enzyme, okay? So if I go back to my same schematic, if this is a diseased cell with these 2 non-functional genes, now we have to use multiple viruses to transfer a number of different functions into the cell. And actually, again, instead of adding another gene in there, we're just correcting this one mutation, okay? So what's up with these guys? So this is the schematic showing the technology, and we call it CRISPR-Cas9, those of us that live our lives in the lab. And it's really not that different from zinc finger nuclease-mediated repair or gene editing, except that it uses a different complex. It uses what's called a Cas9 complex, which is this enzyme complex here. But see the little green stuff in there? That's actually a piece of RNA. And if you remember when I talked about the zinc fingers, we can engineer zinc fingers to scan the genome, and find a specific site, bind it, and then cut it. Well, in this particular case, what we call a guide RNA, that's what scans the genome. And it finds a particular spot that you have engineered into this guide RNA, and then it binds that area, and then this protein complex cuts the DNA, just like the zinc finger nucleases, and you need two viruses to deliver this whole mess here. And then you need a third virus to have this repair DNA also, because when you cut the DNA, just like with the zinc finger nucleases, your cell wants to repair that, and when it repairs it, it can use this as a template and now insert this correct portion of the gene into the area where the mutation was, and you wind up with a correct gene, okay? So, why gene correction? Again, this is really the holy grail. This is what we would love to accomplish. This is exactly what I described before. These are the gene replacement vectors I told you about with the insertional mutagenesis issue. If they jump into the wrong part of the genome, they can cause problems. So this is the problem. With gene correction vectors, again, we're not putting another copy of the gene in there. Hopefully, we're just correcting the gene that's there. So again, this should reduce the chance of harmful insertions in other places in the genome, okay, at least in principle. That's what we'd like to accomplish here, okay? Currently, there are no clinical trials underway yet. This technology really is in its infancy. We can do this in the laboratory setting, we can do it in cells. It's not ready for prime time yet, but there's an enormous amount of potential here. Again, the holy grail of gene therapy would be to correct the defective genes that are in there, okay? So, to close up, what'd I do? Okay, so to close up, so what are the strengths? Gene therapy has an enormous amount of potential. One of the strengths is, at least in principle, this could be a one-time treatment. You take your child in, you have the gene therapy procedure, that's it. You don't come back. You don't come in weekly for enzyme injections or every other week. Potentially, it could be permanent. Also, if things work well, this would provide a persistent source of the enzyme. Again, you don't have to come in every week. Persistent source. And again, if we can regulate the levels of enzyme activity to have them either in the normal range or at least within a therapeutic range, then, and if that lasts for the life of the patient, you're in great shape. So these are the strengths. So what are the challenges that we have ahead of us? Efficiency. Right now, again, this is why I reviewed cross-correction. We do not have the ability to genetically modify every cell. So we still have to rely on correcting a minority of the cells in the body and hope that they will secrete enough enzyme to give you a systemic response. Okay? So efficiency is an issue. Irreversibility. One of the strengths is that you only have to do this once. It also could be a detriment. Unlike enzyme replacement, if you're giving your child enzyme replacement and it has an adverse reaction, you stop. It's pretty straightforward. This is irreversible. Once you inject those viruses, you can't take it back. You're done. That's it. Which leads to the safety issue. Although there's been probably thousands of experiments using gene transfer vectors in animal models, and in general, these things seem to be pretty safe. But recently, there's been several publications showing that there may actually be some acute and chronic toxicity associated with some of these vectors. Okay? So safety is a concern. Safety and irreversibility. Now, I don't want to end on a bad note here. This has an enormous amount of potential. And again, the data up to this point, in general, these things look pretty safe. Okay? But we still have to do more research, more development to make sure that they, in fact, really are safe and that they can accomplish the goals that we want, which is to have the patient make their own enzyme persistently and make enough enzyme to be therapeutic. Okay? So I will also say, I had a half hour to go through this. If there's anything that you didn't understand here, I wander around here aimlessly for the next couple of days. If you see me, grab me. I'm happy to talk to you about it. If this wasn't understandable, or if you want more details, happy to talk to you. There's also other experts in gene therapy here at the meeting. I'm sure they'd be more than happy to chat with you as well. That's why we're here, to help you. So with that, I'll stop. And I'm over time, so I don't have time for questions.
We've got time for a couple of questions.
Hey, how you doing?
Does this solve the blood-brain barrier issue?
No, not at all. The blood-brain barrier is, we're all alive today probably because of the blood-brain barrier. It keeps toxins out, it keeps viruses out, it keeps bacteria out. And you want to protect your brain. So we're probably alive today because of it. But it's also a detriment. Again, the simplest way of gene therapy is hold out your arm, you stick a virus in there, you now start expressing enzyme. And it's really quite effective. I mean, we've we've very effectively treated several of our mouse models where they look essentially normal. The enzyme will not cross the blood-brain barrier. The virus will not cross the blood-brain barrier. It is probably the two major challenges that are still facing us with these diseases are the CNS disease, so the brain disease, and the skeletal disease. And the brain disease is tough, and that's why we and others have done experiments using direct delivery of the virus to the brain, okay? It sounds gruesome. It's not the preferred way to do it, but the blood-brain barrier is a problem. It's a real problem. Yes?
How do you look at the recent Nature publication
Well, that's why I added the safety thing at the end here. So the question is the CRISPR technology. You know, again, on the surface, this looks ideal. You engineer these guide RNAs, you engineer these proteins so that they very precisely scan the genome and pick one spot and either correct the gene or replace the gene, one or the other. But there are data coming out, and again, that's in a perfect world. The data that are coming out would suggest that, in fact, two things might be happening. There could be what we call off-target events. So in other words, instead of precisely going to that one spot that we've engineered, it may actually be going to additional spots around the chromosomes, around the genome, and causing problems there, deletions, insertions, that sort of thing. There's also evidence coming out that when these processes happen, so when you cut the DNA and the repair enzymes start going, that actually could be a signal for that cell to die. Okay, so and that's why I tried to stress that this gene editing stuff, it's really in its infancy. And I wanted to bring it up to let you know that there's an enormous amount of potential out there, okay? But we don't understand it well enough yet to say, let's go to the clinic and let's try this. Because again, it's irreversible. And I think we really need to understand what's going on here first before we jump off that cliff, okay? And I don't know if that answered your question, but basically, there's things we don't know yet. We have to understand them before we, again, jump off that cliff, because you can't climb back up. Yes?
Time for one more question.
You mentioned that not even the virus nor the enzyme would be able to cross the triple B, right? But I mean, if it's being injected into the brain, would it be able to go through to the other side to hit the body?
So that's a good question. And we and others have done lots of experiments where we've injected virus directly in the brain of, say, a mouse or something. And when you do that, if you think about it, again, it sounds kind of gruesome, but if you think about it, if you're putting a needle in, you can't avoid hitting some small vessels as you're putting the needle in and you're injecting the virus. What we and others have shown is that there's actually quite a bit of virus that will leak out of the brain as you're doing that. And we've seen some cases where there's enough virus leaking out where you actually will get a systemic response as well. Now, systemic meaning liver, spleen, heart, that sort of thing. Now, I think, do you want to rely on that to treat the cardiac disease or the liver disease? Probably not, but there is certainly some benefit to the rest of the body by doing this, just by the fact that these viruses can leak out as you go through vessels and stuff.
But not vice versa?
No, not vice versa. That's the problem. So if you do an injection in your arm, in the vein, it's going to help the body. It's not going in the brain. Okay. Thank you.
Thank you very much. Now, our next speaker is going to be Chet Whitley. Is Chet in the room? Ah, there you are. There you are. Chet's well known around here and around the world, and Chet is an MD and is tenured professor at the Department of Pediatrics, Department of Experimental and Clinical Pharmacology, the director of Gene Therapy Center, and an Advanced Therapies program at the University of Minnesota. He's also been involved in quite a number of programs to do with MPS and clinical trials, and is one of the main reasons for the hugely successful World Symposia. So over to you, Chet.
Why, thank you. And everybody, do you want to stand up and move around? I'm sure you're kind of, that's the spirit. Get up and move around. So they got the water up here so nobody can throw it at me. Okay, what else, if you want to move around? Ramsey's going to play some music while we're getting going here. Cue it up, Ramsey. If not, we'll just have to settle for the slides here. So I'll try to move this right along. I know you've been here a long time. Hey, there you go. That's good, right? Now, the scientist people know something very interesting is going on here, but not over there. So let's go ahead here. I'll tell you about a few things. Let's see, this button doesn't work. What do I have to do special?
There you go.
There you go, okay. Well, I was reflecting on this 15th International Congress, or Symposium on Mucopolysaccharidosis, thinking back to the very first one, and some of you may not know this, so I thought it was worth mentioning. The old president of the National MPS Society, the National MPS Society started in the U.S. in 1974. And by about is Joan Cohen here somewhere? Joan, okay, correct me on the dates. So when did you join the National MPS Society in New York City? 1986. And about 1987, Maria Capobianco came to me and said, hey, Chet, we got about $3,000 or $5,000 somewhere in that range to spend on research. And I said, Maria, nobody in the lysosomal disease world is talking to each other. We could do a lot. And Michael Beck, who's here, I think, isn't he? Michael? Maybe he's next door, said, yeah, that's true. And so I said, Maria, if you give me that $3,000 to $5,000, I'll figure out a way of getting about $30,000, and we'll organize the very first international symposium on mucopolysaccharidosis and related diseases. And of course, Christine Lavery from the UK said, hey, this sounds like a great idea, Chet. Why don't you call it a Congress? Because I'm British, We'll call it a Congress. And so we changed the title for the first meeting. But we had a great meeting in 1988. I think I see Joe Munzer over there. He was there. And we had people like Robert Gorlin, Bill Crivet, Pierre Marteau, who, of course, with the LeMay husband and wife combo, discovered Marteau-LeMay syndrome. We had Victor McCusick, who was officially the father of medical genetics. He defined and categorized a lot of the mucopolysaccharidosis and other diseases. He is the reason we now have Mendelian inheritance online, so you can see all the genetic diseases. Elizabeth Neufeld, who's here at the meeting. Sylvester Sanfilippo, Asai I call him because he was a pediatrician in my department. He was in the 1960s doing a fellowship with Robert Good, the guy who discovered B lymphocytes, and said, why don't you look around to these MPS patients, published some of them, and that's how we got to know Sanfilippo syndrome. And of course, Harold Hank Shea, who had been a University of Minnesota graduate student, went out to University of Pennsylvania, and found the first patient with MPS 5, as they called it back then, Shea syndrome. And that is pronounced Shea, by the way, Shea syndrome. That's what he called himself. In any case, that was the first International MPS Society meeting, and it's progressed, and you know the history after that. So I have disclosures to make. There are many people that contribute to the work that I do and the work that people in our group do. And one of those important ones is the National Institutes of Health, who funds the Lysosomal Disease Network. And I hope that all of you will go to the website, lysosomaldiseasenetwork.org, because that's where we fund this, where this funded NIH work is going on. We try to involve patient advocate groups, academicians, of course, pharma, foundations, to advance on our understanding of lysosomal diseases and work actual clinical research. We're coming up on our third annual renewal for 5 years, $6 million of work toward getting people together, talking about lysosomal diseases and doing research, learning new things. So this is actually the organization of the Rare Disease Clinical Research Network. It falls under the NIH, a group called the RDCRN, Office of Rare Diseases and NCATS. And each one of these disease groups has its own consortium to do research on rare or orphan diseases. Importantly, here's the Lysosomal Disease Network. All of us must collaborate and cooperate with respect to a data management and coordinating center, so our data goes into a public warehouse where it eventually becomes available to the public, as well as a coalition of it's actually now more than 100 patient advocate groups, advocacy groups, like the National MPS Society and some of those extra U.S. Groups. So it's an interesting phenomenon funded by NIH. As we think about the lysosomal diseases, some people categorize them as many as 70 or more. It depends on the gene or the name or how you want to discuss this. And we'll put this on the website if you really, really want to see all this. You don't have to take photos if you want, but go ahead if you want. Importantly, I've looked at therapy for lysosomal diseases over the years and divided up into these categories. The era of discovery of cross-correction, largely the work of Elizabeth Neufeld and her colleagues at NIH, as well as Bill Sly. Once we discovered that lysosomal enzymes could transfer between cells and still be effective, we started working on enzyme replacement therapy. The actual first attempt at a human enzyme replacement therapy is when Roscoe Brady flew to Minnesota and tried to give a child with Sandhoff disease human enzyme he had purified from urine. That was in 1980. We've come a long way. The first drug was approved, Ceridase from human placenta, in 1991. And that's why people with Gaucher disease are surviving longer. Then we've looked at small molecules, the first being in 1993, combining different therapies as we do for cancer and other drugs like stroke and heart attack. Synthetic enzyme replacement, that began essentially in 2015, and now the era of gene therapy. So I won't belabor this because Mark and others have talked about it. Cross-correction, receptor-mediated exogenous enzymes into cells and localizing these to the lysosome where they belong. I did mention that Roscoe Brady pioneered this field of enzyme replacement therapy. This is why we also have to think about identifying patients very early, like newborn screening. In Minnesota, we started doing bone marrow transplants, and Elsa Shapiro and I and others learned that IQ drops in a child with Hurler syndrome 20 IQ points per year for every year that therapy is delayed. 20 IQ points per year for every year a child is not treated. We started newborn screening last year in August of this year. We found our first patient with MPS I. I can't take any credit for that. That's the state of Minnesota newborn screening program. But it will be the tidal wave that sweeps across the U.S. There's now 8 states screening for MPS I and for Pompe disease. So bone marrow transplant's very important for what we're going to talk about, which is gene therapy. I did the first bone marrow transplant in the U.S. after Jack Hobbs in London published this in The Lancet. He did a bone marrow transplant for one child with Hurler syndrome, said it was a cure, and it was published. But frankly, nobody believed him. Bill Crivet, my colleague, my boss at that time, did a bone marrow transplant for a child with Bartholomew syndrome. That went well. It saved that child's life. That young lady was having obstructive airway disease, which could not be treated surgically, and she would have died within a year. But she lived many years after that, went to college. She actually died last year. Considering that was 1982, think how long she survived. So my job, when Bill hired me, was to say, can we really confirm or refute that Jack Hobbs can treat Hurler syndrome with a bone marrow transplant? And the rest is largely history. This was actually the first effective systemic treatment for lysosomal disease, hematopoietic stem cell therapy or bone marrow transplant or umbilical cord transplant, and it's what we now use as soon as we identify a child with Hurler syndrome. It prevents the death of the child, and it also protects the brain, much to our surprise. So this really led to, in many ways, the era of enzyme replacement therapy with Gaucher disease, alglucerase, and imglucerase. All these enzymes have gone through FDA clinical trials and are approved. And in fact, the next one this past year is not on that list, the ultragenyx drug Mepsevii, which you'll see about here if you talk with the ultragenyx people. But actually, when you think about it, the term gene therapy was coined by Ted Friedman a long time ago. The concept of snipping genes and moving them around led to the idea of gene therapy. And we now know that is an active therapeutic endeavor. In fact, the very first gene therapy experiments were often using a virus to move a gene from one place to another. And that always, to me, was a fascination. And in many ways, that's how my interest in this field and therapy has developed. So the NIH keeps track of all the human gene therapy experiments done around the world. This focuses on those for lysosomal diseases like MPS. The first one in 1990 was actually the landmark, the first time that Mike Blaise French Anderson actually treated a child with a boy in the bubble disease and put the normal enzyme into a few of their lymphocytes, which circulated around and around and around. It was presumed to work only for a short period of time because of the way they did it, but they discovered that some of the cells in two of the 3 patients, or one of the 3 patients, actually survived a very, very long time, as though they had hit a bone marrow stem cell by mistake. But it led to that child's long time survival and being largely disease-free. Now, there were a lot of other gene therapy attempts in the interim, but 3 of them in the 1993, 1984 era were based on the first experiments in gene therapy. And so at NIH, Carlson, John Berenger at Pitt, and then Robert Schuning, actually he was at NIH back then, and Schuning tried to put bone marrow stem cells back into a child or a person, rather an adult, who had been transduced with an MLV, Maloney-Murine leukemia virus. And the problem back then was that the bone marrow stem cells did not divide. They did not go through cell division. And that's the problem, because you want those cells, those key cells, those are the stem cells we use in bone marrow transplant. You wanted them to be corrected and survive for the rest of that person's life. But we did not have the technology really to do it very well back then. We took, in 1995, lymphocytes, used the same kind of vector, and tried to treat a man with Hunter syndrome. Jim Oliger from the Quad Cities in Iowa came up, donated all of his blood for, well, his lymphocytes for his first visit. And then every month he would come back after we put that same virus with a normal hunter enzyme gene into those cells, and we gave them back to him. After doing that for 12 months, 12 infusions, no effect. At least nothing that we had good endpoints to measure, so we stopped. And Jim was really iconic in the mucopolysaccharosis world for being the first human volunteer. He survived many, many years. In fact, he just died a few years after that because of a large glycosaminoglycan obstruction in his spinal cord. So if we can actually access central nervous system disease, remember, we're not just trying to prevent cognitive deficits, we're trying to prevent hydrocephalus, we're trying to prevent obstruction of the, or actually compression of the cervical cord, so we're not paralyzed. So the central nervous system is not just cognition. There are many other aspects of the central nervous system we're trying to treat or prevent from becoming bad with any kind of therapy. You can see that as time has gone by here, AAV vectors have been very, if you will, popular, and that's because of their perceived safety. They're relatively non-immunogenic, and I say relatively. They're non-dividing viruses. The initial thought was that we could target a particular chromosome with an AAV virus because that's what the normal AAV virus does when it causes the common cold. However, as soon as you start modifying that vector, that virus, take out the reproduction genes, it actually loses that ability to integrate into chromosome number 4. So now we have a virus that does not go into the nucleus, does not go into the rather it does go into the nucleus, but it does not integrate itself into a chromosome. So many people like that. It's less likely to be breaking or interrupting other genes. And so it's been very common. And right now, it is very difficult to find any kind of production facility that will make AAV viruses because there's such a huge demand. There's a huge waiting line for companies and even academicians to produce enough good quality AAV vector to try all these experiments. That's how popular it is right now, and that will change. One thing I want to note, and I'm going to note this a little bit later, but look at Ron Crystal at Weill Cornell Institute. He's part of the Lysosomal Disease Network. He's been using AAV viruses, injecting them right into the brain of children with Batten disease since 2004. That's 14 years ago. So if you want to know what the risk of drilling a burr hole and putting a tiny catheter into the brain is to deliver a vector, there's a long track record. If we go forward, we tried to do that. We got what am I trying to say? A RAC approval to do that in a patient with Sanfilippo syndrome for various reasons that did not work out, but now that is coming to the U.S. Under the umbrella of Lysogene, and I'll tell you about that. And the company Regenix is also using AAV for intrathecal administration. So there are many things in progress. I'll kind of go over these very briefly if we have enough time here. So notice all of these, Sanfilippo syndrome. And many of you understand that there is no effective treatment for it right now. I supervised a 25-patient natural history study that Shire was doing for enzyme replacement therapy. It was great data, wonderful project, very expensive, ran for 3 years. We tracked IQ and other things for 2 years on each patient. And there were patients still coming to us after the natural history study was done because we had developed a good standard of care, if you will. Shire then launched its intrathecal enzyme replacement therapy, and we learned last year that did not meet its endpoints. So even though it might have been effective, we did not have the measurements to determine that. To do a clinical trial, to get something through the FDA, one has to have the right criteria and a long enough period of observation to convince ourselves and the FDA that we really have a viable treatment. So gene therapy has become very interesting, and several groups, and we'll talk about a little bit more of that as time goes on, have focused on Sanfilippo syndrome. I'll talk a little bit about this is one attempt, but I was going to talk about something else here in a minute. In any case, early on, one of the first attempts to directly treat the brain with gene therapy in human beings was published by Marc Todieu and the group out of Paris using the Sanfilippo A gene and sort of an additive gene. It's something that was hopefully going to amplify enzyme production. 4 children were treated. A company was formed around that therapy by a woman who had a child with that disease, and that's quite remarkable. That's now Lysogene. We also want to give brief mention here to Regenix and their attempt to develop intrathecal AAV enzyme replacement therapy for MPS I. The first specific example now available and recruiting patients, many of you know about. A company, Abeona, is working primarily with National Children's Hospital to work on gene therapy for Sanfilippo syndrome type A and type B and a few other conditions. And I probably don't need to tell you this background information they've provided. And thank you to the companies that I'll show you who provided this information. Let me talk about their studies. But they are right now in clinical trials. They talk about the fact there's several variants of AAV vectors. So one can change the outside of the vector artificially to create a variant of the AAV virus, like AAV1 or 2 or 3 or 4 or 5 or 6, so that they have different targets. So the most common vector we see in the environment normally just goes to the liver and the heart and the muscle. But various ones, such as 9 in particular, seem to be tropic or intrinsically able to go for brain tissue. So people have used these as criteria for designing their therapy. It's not something that you as a patient, frankly, should give a lot of concern about, because the company spends millions of dollars trying to figure that out. They're not going to launch a clinical trial unless they have one heck of a lot of data to say it's got a good chance of working. However, it's good to know about that. So this is the impact in an experiment done by people associated with Abeona to show that type 9, when it's injected into an animal, seems to get more of this biomarker, if you will, this artificial biomarker called a green fluorescent protein into the brain in comparison to other serotypes. So that's how people try to tweak the system to optimize delivery to whatever tissue they're trying to get at. That's one factor. When they do this, and this is very interesting because they're considering both intravenous, an IV infusion of vector, and also going to the cerebral spinal fluid, and they've done some experiments that move them in one direction. One of the other things that they did to optimize their therapy is by using what's called a self-complementary AAV. So the genome, the genetics of the vector folds upon itself. It's more stable. It survives longer. And it may get as much as 10 or 100-fold more DNA active in the nucleus of the cell. So this is the kind of biology that's going on in the background before a clinical trial is launched. Right now, they've settled on intravenous administration using that AAV9 vector as the route and vector of choice for their whole, what they call a platform, the series of drugs they're trying to develop. And these are the reasons why. They demonstrate that CNS pathology is reduced in the animal experiments they've done. Not only do they get the brain when they give it intravenously, but they inadvertently also hit some very other good places, like liver, spleen, et cetera. So yeah, their target is the brain, but they actually wind up giving a pretty systemic therapy. Here's some studies they've published, giving that to an animal. So that happened to be a monkey, by the way. They've done this as most FDA studies are required to do, FDA-reviewed studies. They give in different doses. Here are like 5 times 10 to the 12th vectors in the first group of 3 subjects, 10 to the 13th vector genomes per kilogram body weight in the next 3 subjects, and then 3 times 10 to the 13th viral genomes per kilogram of body weight in the next 5 subjects. So you can see they start low doses, go up. That's pretty much what every phase 1 study does. Sometimes phase 2, look at what different doses give you some effect and helps you determine is there any toxicity at the highest level. So once you get through phase 3, you have a pretty good idea about what's safe and maybe a hint at what's effective. They're currently enrolling additional patients in all 3 global sites in the US, Spain, and Australia. So these are some results. They've probably presented those or would be happy to tell you about these because these are now public data, but they show a durable time and dose-dependent reduction in cerebral spinal fluid heparan sulfate. That's one biomarker or one chemical we measure in the CSF to try to get an idea, even though we give an IV drug in the blood system, can it get across the blood-brain barrier or at least have a metabolic impact in the brain? And we can't measure the brain by biopsy, although that was proposed yesterday in a committee I was meeting with. But the cerebral spinal fluid obtained through a very simple, very safe procedure called a spinal tap measures the bad stuff, the heparan sulfate, and if it goes down, that means it's probably going down in the brain. The enzyme is probably working there, even though it's given with the vector intravenously. Magic, huh? That's the kind of magic people are trying to create. So this is just their dose response, if you will. In the first low dose group, the cohort 1, then you can see in the green lines there, they're about the same or going down, maybe a little bit better. And then cohort yellow, this is cohort 3. You can see the effect. These bars, these things called whiskers, you should get used to. Are they irritating? Does anybody know what those whiskers are? they give you what's called a confidence interval, or maybe a standard error of the mean or standard deviation. It's a statistical measure of how different this is from that. And because these two whiskers overlap each other, these are really no different to a biostatistician. You look back and you say, oh, there's a trend here, and we look for those trends. But with only 3 patients or 5 patients in each cohort, you can't do the statistics to say this did not happen by chance alone. That's statistics, and it's really important because that's why Shire failed in their intravenous drug therapy. That's why Amicus failed with a very good drug called Galafold. They didn't have statistical significance. Anytime you're doing a clinical trial, you've got to be convinced. And the best thinkers on this look for what's called statistical significance. That's what those whiskers mean. So here's more information. They're talking about the natural history study. They've done some liver volumes, in this case, looking at the fact that they go down in compared to normal down here. They're still high, but they're going down toward normal, so that's a good sign. And they're looking at the deep brain architecture stabilization, the thalamus volume, a part of the brain, that the treated patients, on average, improve or at least get bigger over a 12-month time period, whereas the control group went down. So this is implying that the thalamus degenerates or gets smaller if there's no treatment in patients under a natural history study. But is stabilized or goes up if you give a therapy. Again, look at the whiskers. Well, they look pretty convincing. They do not touch each other right now. That's a good sign. So I guess I better go back here when I hit the button too quickly. Also looking at the putamen, you see that untreated patients on average go down. There's a range here. That's what these whiskers mean. But that the treated patients don't go down as much. That's a good sign. So caregivers, parents, were also asked to make comments, and they made these comments. This is great. It's supportive information, but it's not going to get a drug through FDA approval. You've got to quantify it and make sure it's statistically significant. So in summary, they're concluding at this point in time, well-tolerated drug is given to 11 subjects. There's evidence of efficacy, including decreased CSF gags, which some people have some concerns about. Is it really a reliable indicator of cognitive function or injury to the brain or not? Smaller liver volume, decreased urine gags, including heparan sulfate, and preliminary evidence for stabilization or improvement of some cognitive functions at 6 months after treatment in just a handful of people. Remember that. It's a very small number of people. They're going to dose cohorts 2 and type 3, and they're showing a time and dose-dependent biologic effect. So this is a further demonstration of some things that they're looking at. They're very proud and happy with, so far the results are going in the right direction. So let's switch gears. Regenix is a company that is very proud about its NAV technology platform. And what that means is they've made a bunch of vectors, AAV vectors, that have different kinds of coding on the outside. They're trying to custom tailor different AAV vectors for delivery and for certain purposes. And they've published it in New England Journal of Medicine. They've published it in Nature Biotechnology. And maybe I should highlight a few more things. They say they get higher gene expression in certain circumstances. Some of them give you longer term gene expression. They have broad and novel tissue selectivity. So some of them are designed, or at least turn out to go to certain organs, others go to different ones. They seem to have a lower immune response if they tweak the outside of that vector, and they are maybe improved in their ability to manufacture. So commercially, these are a panoply of vectors that you can use to use custom, for certain purposes. So as I said before, the AAV vector is the one we find around in nature. They've got one called AAV8, which they think gives 10 to 100 times greater gene expression. That's a good thing for them. They make less drug. A smaller amount of drug is more effective in making enzyme, presumably. They're looking at more efficient gene delivery to sites of most retinal dystrophy. So this particular one, 8, was used here in the retina. Untreated retina. This is a retina which is not nice and healthy, got more green cells there on the surface, more photoreceptors, and is interesting for disorders of the retina. This is kind of an illustration of all their vectors. It's not a complete catalog. They've got over 100 novel or different surfaces on these vectors, so that's what they brag or boast about. So how do they apply it? Well, they're into the lysosomal disease model. If a vector, an AAV vector that they've customized, is combined with a gene like the Sanfilippo gene or the Hurler gene or the Hunter gene, they put it into a cell, and it is used to affect or transduce a cell. And then, of course, not only is this cell now making the normal protein, the missing protein enzyme, but it's able to leak it out and go to other places. So wouldn't it be nice if you could make, maybe 5 or 10% of the cells of the body making normal enzyme, and it leaks out to the liver, the brain, the spleen, the bones? Wouldn't that be nice? That's the goal. You all know that. A single transduced cell has the potential to correct many other cells around it. So right now they are in phase 1 studies for mucopolysaccharidosis type 1 and mucopolysaccharidosis type 2. They've just opened these studies. They call them RGX-111, RGX-121. You can find them on clinicaltrials.gov. You can see me if you want to be enrolled in them because I'm involved in this. They have all these fancy things like orphan drug designation, rare pediatric disease designation, fast track designation. These are regulatory things that it's easy to apply for. They brag about when they get it, but it makes them get through clinical trials faster, and the investors like that because they get their money back real quick, if it works. So for treatment of mucopolysaccharidosis type 1, this describes their vector, which they call RGX-111, and they're using an AAV9. Now remember, the previous study used 8. This one is 9, which is supposed to be what we call tropic for the brain. And the gene they're going to put in is the Hurler gene, the iduronate gene. And they're doing a very interesting thing. They're switching up the route of administration. Remember, the previous study, Abiona, is going intravenously. These guys are going to go straight into the brain, or at least very close to it. Now, Mark says, yeah, you don't want to put an AAV vector into brain tissue because it could develop immunogenicity, and you're going to get necrosis if the brain cells, are what we call displaying a different epitope and therefore get destroyed. So what they're doing is picking a very interesting and safe place right here at the top of the spine, right under the neck. And although I worry about people with MPS conditions having unusual anatomy in that area, it actually probably is, under the right circumstances like CT guidance or MRI guidance, safe to put a needle right underneath the skull into that relatively empty space right here and administer vector. Well, then it can go around to the brain and get to the brain tissue without having to go through the blood or other places. So that's what they're working on. When they treat, in this case, cats, and I think Mark Haskins probably had a hand in this, they can show that there's substantially greater central nervous system correction than that observed in those treated with bone marrow transplant. So they believe by doing this procedure in a cat, they get better effect than they do in a cat that's gone through bone marrow transplant. And this is an interesting piece of information here. Remember when I started, I said, we're interested in the cerebrum, the thinking part of the brain. But there's blood supply in the brain, and there's a lot of mucopolysaccharide in those blood vessels. And it gets to be so big, you can see them on MRI or CT scan. They're causing a problem. I don't think we understand that very well. Also, the meninges, that coating of thin material around the spinal cord and around the surface of the brain, that accumulates a lot of mucopolysaccharide. And that's why many people with MPS conditions have cervical cord damage, and they have to have a cervical cord decompression to survive and not be paralyzed. So in the interest of time, I better move along here. They're doing similar, they're talking about their primary determinant, their secondary outcome measurement. They're going to enroll up to 5 subjects. And this is what their plan for treatment is over time. And for MPS 2, they're doing something similar, the same site of injection, the same AAV9 vector. They just put in the Hunter gene and try to get the same effect. And they've shown in animals, MPS 2 mice, they get an improvement. Now here's where the whiskers are really different, right? For GM ganglioside, which is ganglioside, which is a biomarker of bad brain, the different doses have increasingly better effects. So again, they have sites in the U.S. and they're open to enrollment. For Hunter syndrome, they're going to enroll up to 6. That's what the time plan is. Now, Lysogene, I mentioned earlier. This is a French company that treated 4 patients. Their route of administration is yet different. They're even going more directly to the brain, not through blood, not through the neck to the spinal cord, but by drilling these burr holes through the skull directly into the brain. And they've treated 4 patients so far. That was 6 years ago. And they've not seen any damage from those injection sites. So now they're ready to come to the U.S. And I think yesterday they had the announcement on clinicaltrials.gov that they're opening for enrollment. And Ron Crystal, who has what did I say, 14 years of experience doing that kind of procedure for Batten disease is going to be the primary site to give their vector that way. So this is very interesting. It's another approach. They're starting with MPS IIIA, Sanfilippo syndrome type A. So if you have interested in that, that is now open for clinical trials. This just shows they're putting a tiny little hole here, if you will, in the skull, inserting a needle through that, actually doing it 6 times so you get both sides of the brain. They've tested it out in various monkey models before going into humans. They've modified the vector to be even more effective or more efficient in enzyme production compared to the first 4 children. And they're talking about here their patient cohort, how they're going to do the dosing, and the safety data that they have, and the potential efficacy. So that's another study to look at. If you want to see this data, it's publicly available. I think Lysogene probably has presented this already. But this is their name of their protocol, LYS SAF 302. It's anticipated to start in November 18, so it's basically opening now. And they'll have sites in Paris, Hamburg, London, Amsterdam, Houston, New York, Minneapolis, and Orange County. So the primary objective will be to assess the drug efficacy in improving or stabilizing neurodevelopmental status on patients at 24 months compared to the evolution of the natural history study of the disease. So let's go to one more study. And this is done by California called Sangamo. And it's actually the first to use gene editing in vivo, meaning in a being. Not ex vivo like bone marrow transplant gene therapy, but actually injecting the agent right into the bloodstream. So what is gene editing? It's important to think about this slightly differently than the other approaches. The human genome is here. It's 23 chromosomes. Each one of these is a chromosome. X chromosome, Y chromosome for male-female discrimination. And on those 23 chromosomes are 20,500 genes. And if you start inserting them in randomly, then you could hit an oncogene, you could cause cancer. So one of the big safety features, the concept of gene editing to a specific target is really important. I had a patent on using a lentiviral vector, a simple injection IV. We could cure mice. One shot IV. I gave up on that patent when I saw this. The safety here is fantastic, in my opinion. So if you unravel one of those chromosomes, remember that each one of these chromosomes may have a few hundred, maybe has a few thousand genes on it. You want to pick out a single gene and put it somewhere here, but not someplace else. That's what specificity and targeting means. You don't want off-target sites. So the idea of gene editing is to pick up that one very precise site, and we're going to use albumin gene to put your gene and put it no place else. That's what gene editing is bringing to the table. They've been working on this for 20 years. Not the 4-year-old CRISPR-Cas approach, but something that's been known for 20 years called zinc fingers and zinc finger nucleases. So briefly here, the zinc finger protein structure that binds DNA has been known for 20 years. These are proteins, or actually small peptides, right here, these 3 amino acids that bind to DNA in just 3 places. So a zinc finger peptide rather, will bind to this one site. Every place in the DNA where you see a thymine, adenine, cytosine, that's where that peptide will bind. That's what a zinc finger is. It forms a clamp, if you will. This is one part of the clamp. Here's the other part, so to speak, and it just kind of binds to every place you see this sequence, TAG. Well, there's a lot of TAG sequences in the human genome. That's not enough. We want to do something more than targeting to just any specific 3-base pair. So they've made artificial zinc fingers, and they can basically target any site in the whole genome specifically. Here's how they do it. They'll take, say, 6 of these synthetic zinc finger peptides and link them together, attach them to a FOC1 nuclease to generate a zinc finger nuclease. There's one right there. So a pair of these zinc finger nucleases will precisely recognize a unique 36 base pair sequence. It's becoming a GPS, okay? So this zinc finger 1 here, if you put that in a cell, it has to recognize 36 specific ATCGs to go there. Maybe there's 1 or 2 or 10 or 100 in a cell. But they amplify that specificity by simultaneously putting in a second zinc finger nuclease, which has its own set of 36 letter determinants. So it's kind of like telling your GPS to go to Harvard Street and Washington Avenue. How many places, even though those are common names, how many places on the globe is there a Harvard Street that intersects a Washington Avenue? In Minneapolis, there's one. My office is there. But there's probably not too many other places in the world that have a Harvard and a Washington Avenue. So that's where this GPS system, if you will, works. It will only go to the albumin gene the way they've worked on this. In fact, this will only go to what's called intron 1 of the albumin gene. And when it does that, once this one binds here, it sticks there, and the other one binds next to it, then the nuclease can work. Each one of these zinc fingers has half of a scissors. Here's one half of it. Up here, there's a half of a scissors there. When those two bind right next to each other, they will cut the DNA very specifically. And this one's delivered by one AAV vector. This one's by a second one. A third vector brings in the Hurler gene or the Hunter gene or whatever gene you want, and it'll insert it right there. Now, it's very specific. There's two kinds of repair that can happen. If you repair with homology, and we can kind of go into that, but it's a lengthy story. You'll get the donor here. If you do it non-homologously, it'll be slightly different. So you get non-homologous end joining as well. In any case, it's relatively specific. They look at this site very specifically. Albumin, you may or may not know, is the most prevalent protein in blood. The liver makes more albumin than any other protein that winds up in the blood. So what if you could take just a few cells and edit their albumin gene so as making iduronidase or sulfamidase or IDU2 sulfatase and stick it in the albumin gene? Well, yeah, that cell's not going to make albumin anymore. At least one of the genes in that cell is not making albumin. The other one is, but one isn't. Now it's making the enzyme you want. And so you make tons and tons of enzyme instead of albumin. They do this by giving 3 vectors, AAV1, 2, and 3. One carries a left-cutting zinc finger nuclease, the other one carries the genes for a right-cutting zinc finger nuclease, and the other one carries the third gene. It circulates in the blood, it goes in IV over 4 or 5 hours, it winds up in the liver. It only stochastically probably hits less than 1%, maybe less than 1/10% of liver cells, but if it does its magic, it inserts a therapeutic gene into the albumin intron, and that gene is now making enzymes. So we've tested this out for MPS 1 and MPS 2. They came to us actually 4 years ago and said, hey, Chet, we know you've got MPS 1 mice, a colony of them. You know how to do behavioral testing. And so we did the work for them. This is Leo, where are you? There's Leo. He did a lot of this work right here. And this group of colleagues with Sangamo, and we treated mice of these ages. And, several months out, not only are the mice making lots of enzyme, they're clearing their liver, and actually, some we don't publicize a lot, their brain looks clear as well. So MPS 2 is the same story. My colleague Scott McIver, right next to me, has an MPS 2. Mouse County, we collaborate on a lot of these things. This is now published in Molecular Therapy about 2 months ago. Same approach for Hunter syndrome, putting the IDS gene in there, converting a very few mouse cells into enzyme production factories. And here you can see in mouse tissues, 4 months after treatment, liver, spleen, kidney, lung, etc., even the brain has activity in it. If you look at the blood, you get a lot more enzyme once you do this treatment. And if you look at the GAG accumulation on those organs, including the brain, there's some response there. And there's histology to support that. In the wild-type mice you see, this is what a normal liver looks like with a vein here. If you look at an untreated MPS 2 mouse, you see the green arrows? Those are very small nuclei with a big white area of storage material around them. That's the bad stuff right there. You do the zinc finger nuclease treatment, and the bad stuff has gone away. So they look at even things like nerve, and we're not showing brain here, but there's some evidence of brain response too. So now these are open as clinical trials, and these are now open for enrollment. In fact, these are things they'll look at eventually. The first patient was treated for Hunter syndrome with this technology on November 13th of last year. There have been a handful of patients treated, and so far no adverse responses that I'm aware of. So some people say when I interview them for these studies, well, why, if I've had a bone marrow transplant, or if I'm getting iduronidase, would I want to risk this? It's new. It's phase 1. I don't know what's going to happen. This is what I show them. This is my last set of slides. This is a young man, Taylor, for whom I did a bone marrow transplant about 20 years ago. Transplant went fine. And he's transplanted at 18 months of age. 20 years later, he comes back to me and Dr. Jarnes. He's fully engrafted, 20 years of enzyme in his bone marrow, and he has donor levels of enzyme and peripheral leukocytes to that day. That's about as good as you can do for a bone marrow transplant, right? Well, I say, Taylor, how's your therapy going? Are you satisfied? And here's his answer. Well, I'm not as tall as some of the guys in my class. And he's actually a trainer. He is a professional trainer for a women's volleyball team, very active, very physical. He sends me text messages where he's sitting on top of a punching bag doing sit-ups. on top of a bag. 98, 99, 100. This guy's a maniac. He is a real athlete. But, you know, he says, if I could do anything, I'd really like to have looser joints. So I said, let's look at this. I don't think you're going to feel better after one injection. He's kind of needle shy. But when we published this, we had a series of patients, 1, 2, 3, 4, he was number 5, who had bone marrow transplants. And on average, they, before transplant, had about 1,000-fold more GAG in their urine than normal. 1,000-fold. But all of them responded with decreased levels after transplant. They all went down to almost normal levels, like 117% of normal instead of 16,045. So I said, Taylor, we'll give you one shot of Naglazyme enzyme if you want, but I don't know if you're going to feel any different. That was his main thing, looser joints. So we did. Back then, this is now a couple years ago, we said, let's just check your urine GAG level now. Well, yeah, it's pretty good. This is the normal level here. Anything below this is normal. And here he is. And his level used to be, well, way up here somewhere on the ceiling. So let's get a couple, 3, because there's some variation. So let's get 3 levels before we give you that shot of enzyme replacement therapy. He said, okay, but I'm only taking one shot, and then we'll see what happens. So we gave him one shot of galsulfatase, Naglazyme. And then we all went our way. He went back to home. I went on a business trip, came back a couple weeks later. I said, oh, hey, look at that, went down. But frankly, I'm a skeptic. That didn't look like much statistical significance to me, right? So I said, well, get some more levels. And actually, we sent him home with 10 urine cups. And that's what happened. So surprising to me, in fact, shocking to me, and I did not expect this, adding one shot of enzyme replacement therapy moved him from above normal down into the normal range. We said, Taylor, do you feel any different? Well, I don't know. Maybe, maybe not. So it took about 2 years to come back, but he just finished 2 years of continuous galsulfatase now. And I don't know if I have anything to show you on data, but I can tell you it's going to be published pretty soon. He's now consistently below normal. What really matters to him is how you feel. So he and his mom came up to Minneapolis. In fact, we saw him 2 weeks ago. And I said, Taylor, how you feeling now? He says, well, I don't know if I feel any different. Really, 2 years ago, But his mom says, Taylor, what are you talking about? A year and a half ago, we went to Orlando, and, you walked around a little bit. You were in a wheelchair for most of the day. He said, we went back there last month, and you were walking around all day. If you're not doing a lot better, if your endurance is not a lot better, then you got to rethink that. In fact, Elizabeth Broadland, who's here, looked at his heart. It's better. My lesson is more enzyme is better. I was at the American Society of Gene and Cell Therapy, and I had to listen to a bunch of people doing talks on gene therapy. And when I say had to, it really irritated me. And I got up and asked a question. I said, all of the drug trials we do are aiming at our FDA-approved target is minimally effective dose. We want to have a dose of anything, be it gene therapy or enzyme, that has not got adverse effects. If it's got too many adverse effects, that's a negative spot with the FDA. We don't want patients to have adverse effects. So we always aim for the lowest dose that produces side effects and hope we get some statistically significant improvement. That's called least effective dose. Well, unfortunately, it is the least effective dose in many cases. So closing thing, be sure to go to lysosomaldiseasenetwork.org and help us get 5 more years of funding. We're going to involve patients, patient advocate groups, and everybody in some innovative projects. Some of you can do at home on your iPad. And normally I'm in here in San Diego every February because we have this meeting. Well, next year we're moving it to Orlando, and if any of you would like to come, it's open to patients, but it's pretty hardcore science. You can get some of the background on this, but you're certainly welcome. Thanks, everybody.
Thank you very much, Chet. Okay, we're going to take a quick comfort break of literally 5 minutes, and we will restart again at 3:15. Thank you.